Nathan is postdoctoral scholar in the quantum biology lab, works with Philip, and is working with us on one of the Gaia Foundation collaboration projects entitled Cooperative and Coherent Quantum Phenomena in the Life Sciences. But Nathan's job this afternoon, or this morning his time, is to introduce our speaker today. So over to you, Nathan.
Thank you very much. Can everyone see this?
We've got it, yeah, great.
Great, all right, well, co-author of more than 70 peer-reviewed scientific publications with over 10,000 citations. By my estimate, that's 10,160 citations and counting. Professor Ahmad is a legendary powerhouse of her field. She's currently the Director of Research at the Institut de Biologie, Paris-Seine at Sorbonne University, and Affiliate Associate Professor with Xavier University in Cincinnati, Ohio. Over the course of her career, she has supervised more than a dozen postdoctoral and senior research scientists and nearly 4 dozen graduate students as examples of her research legacy. A trailblazer in her field, she completed her PhD at McGill University in Montreal, Quebec, in my home country of Canada, before she went on to carry out her seminal research discovering the cryptochrome photoreceptor in the Commonwealth of Pennsylvania, where I currently reside. She completed research appointments with the Plant Science Institute at the University of Pennsylvania, the Université de Pierre and Marie Curie in Paris, and the Department of Biology at Penn State University before moving on to her current appointments. A longtime member of the American Association of the Advancement of Science, she has organized and helped to chair several international conferences. One can hardly overstate her contribution to the areas of photochemistry and photobiology, where her pioneering works, most famously the discovery of the blue light receptor cryptochrome. So, to provide some insight to the uninitiated, please allow me to provide a brief historical tour. Plant recognition of and adaptation to blue light were recognized by Darwin in the 1880s, and probably much earlier, and the light receptive flavoproteins specifically were proposed as early as 1938 and certainly no later than 1950. Blue light fluorescent proteins were documented in the 1970s and were dubbed cryptochromes in 1979 for their cryptic hidden nature and their common occurrence in cryptogamic plants. However, it wasn't until 1993 that Ahmad and Cashmore were able to isolate and identify the plant photoreceptor cryptochrome, confirming its structure and activity in blue light phototropism. Over the ensuing years, Margaret built up an extensive body of work with her many collaborators, revealing cryptochrome's mechanism of response to light, including the role of biological electron transfer in its oxidation reduction cycle, its sensitivity to magnetic fields, and the action of subsequent steps during its photocycle through photoreduction and reoxidation, not to mention its radio frequency sensitivity. These led to the discovery that cryptochrome produces reactive oxygen species, or ROS, during its photocycle in the form of superoxide, a reactive oxygen molecule with an extra electron. Ever curious and undeterred by conventional wisdom, Margaret pursued investigations into biological roles for reactive oxygen species, not only as unwanted reactive byproducts associated with oxidative stress and aging, but as fundamental signaling molecules intrinsic to physiology and homeostasis. This led to discoveries that cryptochrome ROS production can be controlled by electromagnetic fields, and that the magnetic field sensitive step during cryptochrome magnetic reception, although light activated, occurs during a light independent dark state reaction with significant quantum physics implications. Recently, these discoveries have motivated the theory that quantum mechanical radical pair processes in cryptochrome could help explain pulsed electromagnetic field-based therapies. Particularly noteworthy is the discovery of the paradoxical effect of temperature on the cryptochrome photocycle, where a decrease in temperature can counterintuitively increase the cryptochrome photoactivity. This may be relevant to photobiomodulation therapy, where light can be used to inhibit unhealthy immune responses by living cells by reducing the cytokine production and the ensuing hyperinflammation. Of course, she's not achieved all this by working alone, but rather with the help of many wonderful collaborators, too many for me to list here all by name, from many areas, both geographically and scientifically, that she's asked me to recognize on this slide. Nor is it by mere coincidence, but by cognizant choices that Margaret has keenly and efficiently directed her investigations into the light receptive properties of cryptochrome, its magnetic sensitivity, and now recently pivoting her research program to respond to the global pandemic, transferring her wealth of knowledge and skill from the hidden dynamics of cryptochrome, sensing and reactive oxygen species signaling to work on photobiomodulation and pulsed electromagnetic fields therapies. Therefore, it is a great pleasure for me today to have the privilege to introduce Professor Margaret Ahmad, a pioneer in the life sciences, discovery of the blue light photoreceptor cryptochrome and the mechanism of its response to light, its oxidation reduction cycle, and its magnetic and radio frequency sensitivities. Today, however, she will be discussing the application of light and electromagnetic fields to reduce hyperinflammation triggered by COVID-19, a very fitting and urgent topic considering the previous talks we've heard this autumn series, not to mention the current and ongoing global state of affairs. And so, without further ado, I invite Margaret to take the stage, if you will, and take it away. Thank you.
Nathan, thank you very much indeed. Margaret, just before we start, welcome to Omid and Tristan. I think you're both from KwaZulu-Natal.
Yes.
And I think we've seen Gladys has come on board.
From Paris, yeah.
From Paris, yes, lovely to see you. So Margaret, when you're ready, if you'd like to share your slides, over to you.
Okay, well, thank you very much for that glowing introduction. I'm a bit embarrassed, Nathan. And thank you very much to this really great opportunity to talk here in front of this group. I'm very honored. And so I'm going to talk to you today about how can we use quantum biology to do something about this current global epidemic and hopefully to prevent adverse effects of future ones. So the COVID-19 epidemic is caused by the severe acute respiratory syndrome coronavirus. It's a pleomorphic RNA virus, which has caused multiple epidemics in 2003, 2013, SARS, MERS, and now the one in 2019, COVID-19. And the point I want to make for this whole talk is that the mortality, the actual critically dangerous and life-threatening effect of this virus is not really the viral infection itself. It results primarily because of the effects of the host response, this respiratory distress, which is not due to the virus as such, but due to a hyperinflammatory immune response of the host organism, leading to the cytokine storm in the lungs. So it's really so this is really a step, the mortal step, and it is not a direct cause of the viral infection. It's an indirect result of it. Now, nobody here needs to be reminded of the cost of this epidemic. So far, there have been 5 million deaths. The economic cost is in the trillions, and there is currently no known cure. We have preventive strategies, of course, the vaccines, which are effective. However, they take time. They are not yet available to everybody. They have to be repeated, and also if there's new variants, they may not be effective. As far as treatments are concerned, there are quite a few now: antivirals, anti-inflammatory drugs, ventilators. They tend to be more effective against the early stages of the virus, not so much the late stage. The cons with these current therapies are they tend to be very costly. For example, remdesivir, which the former president received, costs $3,000 for one treatment course. They have many negative side effects, and also they are not highly effective for late stage COVID. So currently, I think it is accurate to say there are no effective treatment options for late stage COVID-19. And in this talk, so the first part I want to explore with you can we use electromagnetic fields for the treatment of this late stage COVID inflammation leading to mortality? So the type of electromagnetic fields that I'm going to talk about are a type which is already currently used for treatment, for the treatment of pathology. And it's called this pulsed electromagnetic fields. And these fields, they're at a very low magnetic field strength in the 2 millitesla range. So they're very weak. They're about 40 times the Earth's magnetic field, 40 to 100 times. And they're generally pulsed at low frequency of from 0 to 20 hertz. So these magnetic fields are generated in the following way. A current is generated by a coil, which is just wound round and round in a circle, a circular coil. And then that coil is placed under the treatment area, which a body part, a cell culture dish, whatever. And a current is applied to that coil, which is generally pulsed. There's a duty cycle involved, and it's a certain current, a certain voltage, and a certain frequency. And as a result of the current, a magnetic field is generated at a 90-degree angle. It's classical physics. And this magnetic field switches on and off at the frequency that the current is generated. So the magnetic field strength is determined by the current and the voltage passed through this coil. And furthermore, another parameter that is looked at is the shape of the magnetic field pulse. So for example, a pulse can be, this is a magnetic field strength of such a coil measured. And this is time in seconds or microseconds. So here you can see when the current comes on, you get an increase in the magnetic field, the current stays on and then goes off. It's a square coil. That shape can be manipulated by changing the frequency and the duty cycle of the electric field. So typically these fields are from 10 to 50 hertz with all different types of shapes. And all these different pulse shapes and intensities and frequencies are varying greatly from one treatment to another and are generally patent protected. So this is considered secret. Every manufacturer, their device. It's hard to get the specs. So these devices are everywhere. They're on the market. They come in all shapes and sizes. There's even wearable devices. I should mention there's quite a large industry for racehorses and animals and veterinarians who use these devices for conditioning of animals. And the treatment dose typically involves 10 to 20 minutes of exposure. So you place this device under the body part or in the animal for 10 minutes a day or 10 to 20 minutes, and once or twice a day, and perhaps for a week or two. So that is the therapy. So where did I get the idea that this might treat COVID? Well, the reason is if you look at the diseases for which PEMF, so pulsed electromagnetic fields, are currently recommended: chronic pain, wound healing, so bone healing, here we are, bone healing, hair loss, lupus, arthritis. All of these are conditions that involve resolution of underlying inflammation. So it rings a bell. In the case of COVID, as I told you, the lethal step is when the virus infects or triggers in the lung the innate immune system to induce a so-called cytokine storm, this step. And these cytokine storms is what induces mortality. So how could PEMF work how could pulsed magnetic fields work to intervene in this pathway? Here I was very lucky in that so much is known about COVID-19 and how COVID-19 regulates the immune system. So very briefly, to give you the published literature on this, it's the spike protein of COVID-19 virus, which interacts with a specific receptor, the TLR4, toll-like receptor 4 receptor of the innate immune system. That interaction, it's mostly this one with a small contribution by one or two others. So that interaction induces triggers an innate immune response, which ends up through a complex set of steps activating this transcription factor called NF-kappa B. NF-kappa B activation goes on to induce the synthesis of inflammatory cytokines. So these so-called cytokine storms, uncontrolled, deregulated, once triggered by the virus, they just go on and on and on and eventually kill the patient. So this pathway is understood, it's known at the molecular level. And I chose an experiment, so my lab, it chose an experimental system where we could actually directly study the effect of pulsed magnetic fields on this particular type of inflammation. And it so happens that you can buy commercially available cells from in vivo gen, which have been engineered to perfectly mimic this innate immune system TLR4 dependent cytokine storm. So you can actually buy these cells. And what these cells do is, what these cells do is they contain the TLR4 receptor, which can be induced, for instance, by the viral spike protein, or in general in the lab, we use a bacterial inducer because we don't want to handle the virus, but it attacks the same part of the receptor. So you can induce the TLR4 receptor in this model cell culture, trigger the activation of this NF-kappa B transcription factor, and result in the production of large amounts of cytokines, as well as a reporter gene that has been engineered into these cell cultures to turn them blue when they're inflamed. I mean, how easy can it be? So we got this cell culture system and we did the following type of experiment. So we cultured the cells, and on day one, we added the inducer of inflammation, in our case, with bacterial inducer. On day two, we started treatments, either pulsed magnetic fields or nothing at all. And we did these treatments for 10 minutes at a time, every 12 hours for 2 days. And at the end of that time, we did a color assay. So these are the cells in a dish with a colored substrate that have not been inflamed. These are non-inflamed cell culture controls. And on the bottom in blue, we have cell culture inflamed, that is inflamed because of activation of this COVID-related pathway. So what happens when we stimulate these cell cultures with pulsed magnetic fields? We used a commercial device at 10 Hertz. So it makes a nice little square wave periodically. And so this just shows you the shape, the famous shape of the signal, and the signal strength determined from the induced electric field. And we placed this under our dish, which we had induced with this bacterial elicitor. We had induced inflammation in this dish of cells. And for 10 minutes a day, twice a day, we placed this device underneath and switched it on. And then after 2 days, we did a color assay, and we could see with our eyes the result, which was that the healthy cells don't turn blue, the inflamed cells do turn blue. But after only 2 days of this treatment, you could see with your eyes that they are less inflamed. The inflammation has gone down. And this is a quantitation of that result. So it's about a 20% decline in this inflammatory marker. So we got pretty excited here because we said, oh, look, we have used magnetic fields and we have cured the inflammation. Cured is not the right word. We have reduced. We have an effect on the inflammation caused by this awful pandemic, which at that time, 2020, was keeping us all locked up. So, of course, when you see an effect like that, you need to understand it. It has to make sense. It can't just be magic. And we wondered a bit about what is the underlying mechanism. As you know, I study magnetic field effects, not so much medical as fundamental effects of static magnetic fields. And that weak signal having this anti-inflammatory effect, it reminded us of what happens in migratory birds. Migratory birds can respond to very, very weak static magnetic fields in the 2 to 50 microtesla range. So it is really the best magnetosensor around is a bird, pretty much. So the question we wondered was, maybe we could explain the mechanism that was going on in the pulsed field response by what was happening in the birds. So it seems a bit of a distance to travel mentally, but in fact, there was a very simple experiment we thought of that we could do. Birds can respond to static magnetic fields. They don't need a pulse, but they can also respond to really, really low ones. So much lower than the Earth strength field. And if you buy commercially a funnel made of mu metal, these are available to anybody who wants to buy them. And in the middle of place your sample in the middle of a funnel, you can decrease the magnetic field just by doing this funnel from 50 to 2 microtesla. So it is a way of significantly lowering the Earth's magnetic field. And we predicted that if this pulsed field effect was similar to what happens in the birds, then this funnel, which lowers the Earth's magnetic field, could have the same effect. So we did the experiment. We took our cell cultures, we put them, we gave them an inflammatory inducer. We placed them in this funnel twice a day, 10 minutes at a time for 2 days. And at the end of this time, we asked, what's the effect on inflammation? And lo and behold, simply placing the plates, these plates in a funnel, was able to decrease the inflammatory response. So this is a control. This is the decrease in inflammation as a result of the funnel. And this is the decrease in inflammation as a result of the pulsed field. So we could get the same effect as for these very fancy pulsed field devices with all kinds of different interpretations of why they work, with simply placing the cells in a funnel. And that funnel was consistent with how birds see the magnetic field. So this again pointed us in a certain direction for mechanism, because it is now thought and theorized that birds use a possible biological magnetoreceptor for orientation, for magnetic field perception, called cryptochrome. And I won't go into this in a lot of detail. Just to say it's a flavoprotein that undergoes certain reactions in response to light and redox potential. So cryptochromes are found in birds, but they're also found in many other organisms, flies and humans. And so the question was, is there a common mechanism of pulsed field detection which involves cryptochrome? That was our next question. This is a schematic representation of the biochemical reactions that cryptochromes undergo, involving flavin oxidation and reduction. I'm not going to go into it in a lot of detail, in any detail here, actually. The only thing I want you to grasp from this diagram is that cryptochromes are responsive to magnetic fields at a certain reaction step, which generates reactive oxygen, ROS. This is a prediction of a mechanism, a quantum physical mechanism called the radical pair mechanism. And it has been experimentally demonstrated for other types of cryptochromes that an increase in reactive oxygen occurs with magnetic stimulation due to cryptochrome. So we wanted to know, does this medical device, does this medical effect also stimulate ROS in human cells, reactive oxygen? So we wanted to know, is it this mechanism? If it is, it should stimulate ROS. How do we measure reactive oxygen in cell cultures? These are cell cultures, a control cell culture, which has been treated with a fluorescent dye that recognizes reactive oxygen. Okay, here we exposed these cells to this pulsed field device. Remember, our question is, does this pulsed field device work like the magnetic field does in birds and in other organisms? So we tried to stimulate it and see whether it made reactive oxygen. This green fluorescent dye went up after only 15 minutes of stimulation. Yes, 15 minutes of PEMF stimulates reactive oxygen in human cells. This is an old study from my lab, but it confirms our thinking. So the next question is, does cryptochrome have anything to do with this response, this pulsed electromagnetic field response that is so anti-inflammatory? And to look at that, we obtained cryptochrome mutant cells. So it's the beauty of working with mammalian cells, is that you have possibility of getting mouse mutant lines. We obtained a mutant line that lacked cryptochrome, and we did the same experiment. We stimulated it with pulsed electromagnetic fields, and you see that there is no increase in reactive oxygen. So, I mean, I leave you with the thought that what we have found is that the pulsed field stimulation, these devices that are being used medically for the last 50 years, they seem to have a lot in common with the way that birds are able to perceive the Earth's magnetic field. They seem to be able to undergo a chemical transformation that generates reactive oxygen. So as soon as you talk about reactive oxygen, you start to think, well, there are many, many medical consequences of reactive oxygen. It could be very toxic at high concentrations, but at low concentrations, the body uses these reactive oxygen species as a means of, and so we asked the question, can this mechanism of magnetic field perception producing reactive oxygen, could this explain the curative, the anti-inflammatory effects? And I hope you don't see all of this. I hope you can see all of this. So very briefly here, again, we have our model slide for inflammation. This is the virus. It triggers inflammation by interacting with the TLR4 receptor. There's a whole signaling cascade down to this transcription factor, NF-kappa B, which releases cytokine storms. But another part of that pathway, as you can see here and here, involves reactive oxygen. And reactive oxygen can actually regulate the activity of NF-kappa B. So if you have a suitable concentration, and this is known, this is published, this is how this pathway works. So if you can manipulate the reactive oxygen in a suitable way, you can short circuit, you can bypass this whole TLR4 receptor stimulation. You can short circuit it so that the NF-kappa B will no longer be super excited and generate a cytokine storm. Of course, we haven't proved it, but at least mechanistically, if a magnetic field affects the concentration of reactive oxygen in the cell, it could be the explanation for why it can treat the inflammation that is caused by these viral binding and this strong inflammatory response. So here's a little bit of a simpler way to say it. And I hope you can see this. I can't get rid of this top of my screen here. This is a little bit of a simpler explanation for this. Here imagine that you have reactive oxygen here, and these are all the different species of reactive oxygen. And the trick about their curative properties is that at high doses, they're dangerous. So extremely high doses of reactive oxygen are dangerous, but at a moderate dose, they are actually therapeutic because they release all the cellular defense mechanisms. They trigger all the anti-inflammatory mechanisms, the DNA repair mechanisms, the lipid repair mechanisms. All the ways that the cell can defend itself against stress are triggered at a moderate dose of reactive oxygen. And this can explain the therapeutic effect of pulsed electromagnetic fields. The trick is to get the dose right. It's very much the trick. You need exactly the right amount, but then you can do a miracle. So from that initial finding, or that initial reflection, and that evidence, which is circumstantial again, we thought to ourselves, okay, if we are right, if this is really the way that these magnetic fields work, can we do something to improve the PEMF signal beyond what is available commercially at this point? Can we improve it? So we made a prediction. Of course, our prediction was yes, there should be a way to improve it. Because first of all, we thought that we need a much more homogeneous magnetic field. These pulsed field devices are not very homogeneous. There's a lot of noise in them. So we figured providing a homogeneous field would help. The second consideration was that if just putting the cells in a funnel was effective against inflammation, could we design a pulsed field signal to mimic a very low magnetic field, which is close to the Earth's field, between 0. So we thought this might be the best range. Let's zero in on this. And finally, because we're going to very low magnetic field strengths, we thought, let's try a signal where we actually take into account the orientation of the Earth's magnetic field. And to do these experiments, obviously, I needed a tremendous technical support. And there is a group, there is a company, SIT Research in Germany, who makes these devices and who does research on them. And they supported these studies. So they came to my lab and they provided the actual magnetic, the signal generator. They installed the boxes, so a control and a test magnetic field device. And inside this device, they designed a magnetic field coil, which was square, so that it could give an even magnetic field signal all across the wells of a Petri plate that was placed inside it. So we have a way to precisely manipulate the field and get it uniform across an entire experiment. So with this device, then we designed, I asked and they did, design a new improved signal. And that was taking into account the following. So we thought, let's design a signal where we take into account the Earth's magnetic field. So the signal was a bipolar signal, where, so here we have time and here we have magnetic field strength, where the current provided a positive signal for about 25 milliseconds. So it went up to 40 microtesla, which is Earth field, for 40 milliseconds. And then the current reversed polarity, and it went down to by 30 microtesla for 25 milliseconds. What I'm saying is the strength of this magnetic field is, so what I wanted to say is the field is going up by about Earth strength, 35 microtesla here. It's going about down by 35 microtesla in the box. And of course, when it's going down, when it's going up, the Earth's field, the Earth's magnetic field cancels it. So we are generating, using this pulsed field device, we are actually generating a pulsed low-level field. And the result of that on inflammation, so we did the experiment on inflammation, okay, inflammation of cell cultures. As I showed you before, 2 days of treatment, 10 minutes per day, twice a day. This is the untreated sample, inflammatory, this is the blue color. And this is the treated sample, less blue, decreased in inflammation. In this case, the decrease was 40%. So we got a much bigger effect than anything we got with these commercial signals, simply by lowering the magnetic field to be within the range of Earth's field and adjusting the intensity in such a way that it could be canceled periodically. So a further point of this experiment is that it's been independently verified in another lab in Tübingen. So we are very confident about these results and very excited, of course, because it is COVID-dependent inflammatory effect that is being treated. So, however, the question is, so to this part of the talk, the question is now we have a signal that could likely be effective against COVID, against this lung inflammatory response. Can it be used? What are the pros and cons? Well, the pros, safe, there's no negative side effects, non-invasive, it's likely to be effective, quick, 2 days, non-invasive, I said, and affordable. These devices, you need one, can treat any number of patients. However, the problem with this is that it takes, we considered this will take a huge amount of time. And the reason is, so I won't go through everything, there are two main reasons for this, one of which is that there's no prototype device ready at this point. You have to design something that will cover the chest area and that will deliver that signal uniformly to all parts of the lung. So it's a bit of an engineering challenge, and it doesn't exist, so it's not ready. And secondly, for these kinds of devices, the number of regulatory hurdles are huge, and that will also take time. So, given that this epidemic is not waiting around for us, I thought at this point to try maybe another alternative. And that one is infrared photobiomodulation. And thanks to the organizers who invited Mike Handlin, who gave just such a beautiful talk a couple of weeks ago on this. So I hope most of you are familiar with this. And this involves using infrared light precisely in the same way to treat inflammation relevant to late stage COVID-19 inflammation, which is where the mortality happens. So could we use infrared light? Now, where did this idea come from? Well, I was vaguely aware of photobiomodulation, but then reading a bit more about it, I noted that every single disease that is recommended for treatment by pulsed magnetic fields is also recommended for treatment by photobiomodulation, by infrared light, just treating with infrared or red light, something like this. Chronic pain, wound healing, bone healing, arthritis, lupus, depression. They're all the same. And all, as I said before, all of these conditions involve the resolution of underlying inflammation. So why not try photobiomodulation? Now, I make a point here. These infrared lights I'm talking about are not heat lamps. You do not want any kind of heating or burning of the patient. In fact, you want the opposite. You want a light output, a type of light which can penetrate into the chest cavity without generating heat. So visible light doesn't penetrate the body at all, practically. That's pretty much excluded. Only that's why I use infrared. Only infrared light efficiently penetrates between 700 and 900 nanometers. So it's really only infrared which is able to even penetrate. Second thing is that the light has to be really powerful. So light intensity decreases with the square of the distance from the light source. So as you go out from the light, it just exponentially decreases. If you want to penetrate the lungs with a useful, i.e. therapeutic dose, you have to consider a light that would still be powerful inside the body from 3 to 10 centimeters from the skin, and think of also the flesh in between. And it should not burn the skin. So that is a bit of a challenge. And I mentioned here also that incandescent infrared bulbs, which are actually really high output, and we tried them first, will not work for this therapy because they heat. They're greater, they're in a wavelength range, which just generates tons of heat and doesn't penetrate at all. So what we used for our study, for our test, was this wavelength here, which is around 750 nanometers. And this is a graphical representation of absorption of light as a function of wavelength into the human body. And the minimum, you see there's a minimum here of most types of absorption. Specifically, we were interested in this one here, which is hemoglobin. So this is the lowest absorption. This is the best penetration of light through hemoglobin. And lungs are particularly high in oxygenated blood. So we focused on this wavelength. Now we went back to our go-to experiment, which is again that the TLR4 reporter hex cell line. Again, to recapitulate, in this experiment, the cells are stimulated with, we used a bacterial inducer, but the virus acts by the same mechanism, which induces the TLR4 inflammation receptor. That receptor stimulates this transcription factor, which then goes on to create these cytokines, which are responsible for the death of the subject of the experiment. So in our experiment, of course, we did cell cultures. We used these cell cultures. We induced the inflammation on day 1 with a bacterial inducer. And then instead of a pulsed field, we used a 730 nanometer infrared LED light. And we gave a 10-minute exposure once every 12 hours, so twice a day for 2 days. So 2 days. And we checked for inflammation using this color assay. And once again, we saw that inflammation was considerably decreased, significantly decreased as a result of this exposure to infrared. So here's a control. The blue color shows inflammation. This is infrared exposed after 2 days. And here's a graphical representation. So this is the control cells, and this is a blue color. This is a measure of the blue color, the inflammatory response. These are increasing intensities of infrared light. So that 0, the inflammation is 100%. At this intensity, we got to a threshold here at 1. We see a decrease of about 35% in the inflammatory response. Remember, it's a model system, but it's only after 2 days and just a tiny little bit of exposure. 6, so what was also encouraging and important was that if we increased the intensity of the light to 6 or 18, so about 20 times the threshold, we got the same, basically the same effect on inflammation. It's an important consideration with light, because when you shine a lamp over a human chest area, you want to be sure that every part of the lung is going to be treated. So every part of the lung will have to receive at least this much light, but other parts of the lung, because the light will come in from one side, and so there'll be a gradient of light from the depths of the lung to the surface. Some parts of the lung might receive a lot more than the threshold. And therefore it was encouraging to see that, okay, we have a threshold, but if you're above that, you're good. No matter how much you put in that system, it will still treat inflammation. A second thing that I noticed, I mean, we did a lot of work on dose. And I'm not going to go over it here because we looked at all different wavelengths. We looked at all different types. We looked at different combinations of wavelengths. We looked at pulses, and we got really complicated effects. I think there's one I would like to draw to your attention because it was really very surprising, and it was the time dependence. So that's in this graph here on the bottom. I told you that we illuminated our cell cultures, which are here. We illuminated them for 10 minutes twice a day. Well, if we changed the time of illumination to 5 minutes twice a day, there was no effect on inflammation. So this shows the inflammatory response with no infrared treatment. This shows the inflammatory response with 5 minutes of infrared treatment twice a day for 2 days. No response. Okay, so that means it's not enough, right? Here is our optimum, 10 minutes a day, twice a day, which is the same as in these upper panel. But surprise of surprise, at 15 minutes, okay, this is just 15 minutes of exposure twice a day. There was no response. And this is very typical of the type of results we got when we tried different conditions. There seems to be a very narrow window in which you get an optimal response. I'm not saying that there's zero effect. There may be some small insignificant effect at other times, but the optimum is really very sharply defined. And so I think maybe one reason why there has been a lot of disparate results and contradictory results even in the photobiomodulation field might be a lack of appreciation of how critical the dose is. Of course, I only tried this one inflammatory response. Now, up to now, we only looked at blue color as our assay for inflammation. And so we thought, let's look directly at cytokines. Remember, we're looking for inflammation as making cytokines which go on to destroy the cells. So in our cell cultures, we did another assay, which was looking at expression of inflammatory cytokine genes in these TLR4 dependent cultures. So I show you the results here, which is, this is a control. This is gene expression in a control cell culture, which has been treated with an inflammatory agent. So it's inflamed. It's making lots and lots of IL-6, which is an inflammatory cytokine, and it has not been treated. Here on the right are the same cells which have been treated with infrared light, and you see a marked decrease in expression of this very toxic, very dangerous cytokine. And we got exactly the same effect for IL-8, TNF-alpha, INF-alpha, INF-beta. All of these are inflammatory cytokines which are induced in these cytokine storms caused by COVID. So all of these are significantly affected, down by 40 to 60%. Thanks to our infrared treatment. And the final thing we did with these cell cultures is we looked directly at IL-6. IL-6 is the most prevalent cytokine induced by COVID-19. This is really the big killer in that disease. So in our cell cultures, we bought an ELISA kit, and we looked using antibody at what is the effect of infrared treatment on the secretion of cytokines, and the result is here. So these are the this is cytokine secretion. These are cell cultures that have been induced with an inflammatory agent, and these are the same cell cultures induced for inflammation but treated with infrared light. You see the change in secretion of IL-6. This is the actual protein made. There is 80% less after 2 days of treatment by this infrared therapy. So it's a very astonishing result, but it's still a model cell culture. It's not the actual patient with the disease, right? So we wanted to take a step closer to the actual situation, and we looked for, does this type of simple infrared lamp LED therapy work to decrease inflammation in actual human lung cells. So in the human lung, there are three major types of cells involved in these cytokine storms, in this cytokine secretion. One cell type are alveolar epithelial cells, which are in the alveoli, which line the lining of the air sacs in the lung. Other cell types important for cytokine in the lung are resident macrophage cells and lung fibroblasts. So we obtained cell cultures, primary cells of these epithelial cells and also resident macrophage cells. We did the exact same experiment. Now, again, we did these experiments in culture. We took the cell cultures of human lung cells. We exposed them to a pro-inflammatory bacterial agent. This triggers COVID-related inflammation. We exposed them day 2 to day 4 to infrared light treatment. And then as our assay, we looked at the expression of cytokine genes. We couldn't use a color assay, of course, because this is not our nice model system. So we looked at gene expression. Here, briefly, the results in the alveolar epithelial cells, non-treated cells, gene expression, treated cell gene expression. It's down. Non-treated cell, cytokine gene expression, treated cell cytokine gene expression. It is down. These are macrophages. These are alveolar epithelial cells. These are the exact same cells that are being attacked in the human lungs by this COVID-dependent pathway. We then looked at a whole panel of them. We did these are 6 of the most critical cytokines: IL-6, IL-8, INF, INFB, INFG, and TNFA. Again, we did this in lung cell cultures. These were macrophage cell cultures. Here we have cells that were induced for inflammation. Here we have cells that were treated for 4 days, in this case, with infrared. Decrease, 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 and decrease in every single case. So it looks very promising from the data, but you're never really happy with an effect until you can think of a mechanism. I mean, how can a lamp, just infrared, just have such a pronounced effect on inflammation? And here, as an explanation, I've lifted a figure from one of the publications where Hamblin is co-author, the review article. So they're looking more at the brain, the effects of infrared stimulation on the brain, but the mechanism is relevant. And in this mechanism that is considered a likely one by the experts in the field, what happens in the case of near infrared is that the photons are absorbed by a mitochondrial enzyme, a cytochrome C oxidase in the mitochondria. And this generates ATP, but it also generates a burst of reactive oxygen. And of course, immediately the light bulbs, figuratively speaking, go off, because we thought, this makes sense. If infrared light can trigger reactive oxygen, we have a mechanism. It would work. So to verify this, we looked in our human macrophage. So these are cells that are actually in the lungs, which undergo these cytokine storm effects. So we looked in these human macrophage cells. And in this case, we didn't look for genes that were affected by inflammation. We looked for expression of genes that are regulated by ROS. So for us, this was to obtain markers for reactive oxygen. You used reactive oxygen regulated gene expression. And these are the results here of a gene expression. So in the control condition, we have human macrophage cells, which were induced with inflammation. These are inflamed human macrophage cells. After 5 days, or we did an RNA extraction, and we looked for expression of these 4 different redox activated ROS-regulated genes. And in the absence of infrared treatment, we got this level of expression. This was the increase. That was huge. That was like 5-fold increase in expression as a result of infrared treatment. It's a 5-fold change in that one. And similarly for all these other genes, control, infrared treated, control, infrared treated, control, infrared treated. Those are going up. In these cases, the expression is down regulated. So control, infrared treated, control, infrared treated. I mean, that's 6 out of 6. It is absolutely clear. You know, there are many more we could try, but I think it's just from this, it is clear that yes, infrared treatment is regulating reactive oxygen, not through cryptochrome, okay? Not through the same mechanism as the pulsed fields, but it is ending up by changing the level of reactive oxygen in these cell cultures, which brings us back to this little cartoon mechanistic slide, which is that if you can regulate, if you can suitably and subtly regulate the effect of reactive oxygen, you can manipulate the body in a positive way. And for inflammation, and also for inflammation at a moderate concentration, ROS can downregulate NF-kappa B and therefore the cytokine storms. So a lot of this is putting 2 + 2 together. I obviously have not, you know, this is work in progress, and we still need to look at every step of the way. We need to look at every individual component to see, is this idea correct? Have we got the right, you know, mechanism? But I find it extremely encouraging that the literature is consistent on this effect. All the literature I've seen on pulsed magnetic fields agrees that there's an effect on ROS. And all the literature I've seen on the immune response agrees that there's an effect of reactive oxygen. And all the literature I've seen on infrared treatment agrees, or at least that has looked at it, agrees there is an effect on reactive oxygen, mostly through this mitochondrial mechanism. So I think we have a reasonable, you know, we can, it's not just that I've observed a phenomenon in the lab, which I like. I think we have a scientifically solid case for a treatment based on these types of signals. So of course, nothing is as good as a case report. And actually I was very gratified, I have to say, that when I did a literature search, I found to my great delight that this idea was not unique to me. It is actually out there. And in fact, a number of orthopedic surgeons, so who use lasers for their practice in orthopedics, had this exact idea back in 2020, even very early on. And they published a number of case reports, which I would like to share with you, on exactly this type of approach to treating late stage COVID. So case report number 1 in 2020 was involved, again, these are all orthopedic lasers, where the exposure was only once daily for about 20 minutes at a time. And the way they do these is they have a small a small window. So they have to pass the laser up and down. It's actually quite labor intensive. They pass it up and down the chest area of the patient for 20 minutes or so. That's the treatment. And so in this first case with a 57-year-old African-American man, the patient improved. So it's a case report, but he got better. Case report number 2, similar kind of treatment. So here they show the areas of the lung that were exposed. Again, it was only once a day. It was it was 4 days, and it was for 20 minutes, for 30 minutes, putting the laser back and forth. But here again, the patient improved. However, what was really, really exciting was they went on, these are the same group, they went on to do a 10-patient small trial. So this time I actually have really a preclinical trial. And again, the same type of treatment, 10 patients were treated. And 10 patients were in the control group that were compared to them. And in the treatment group, there were 0. So they treated patients which were pre-ventilator late stage COVID. So these are patients that had about 24 hours before they were going to be put on the ventilator, they were treated. And they looked at the outcomes of the treatment. So in the treatment group, 0 patients ended up in the ventilator. So 0 out of 10. And of course, that means also that 0 patients out of 10 died. In the control group, which they had matched in every way they could to the treatment group, 6 patients went on to be put on a ventilator. And of those, 4 patients died. So I think, what we can say, at least from this 100% success rate of this sort of preliminary type of effort, is that it's not doing any harm, clearly. I think that that's definitely clear from these studies, and they're very encouraging as far as possible clinical outcomes. So, which brings me back to this global pandemic, which is a very serious pandemic with all these costs and problems associated with it. And what would it mean? What could it mean if photobiomodulation therapy was used for COVID-19? So if proven, so assuming if it were to work, what would it mean? It's affordable. It's because you don't have to use a laser. You can use a LED. We use LEDs. These are not expensive. It's safe. I mean, okay, I think they're not expensive for at least for a test. I think the actual device might cost a bit more, but it's not that expensive. Safe. There are no known side effects. A big one is this: it would be useful against any variant of the virus. So, if we get a mutation tomorrow, it would still cause a cytokine storm. This treatment would still work. Next, and that's another big one, because generally anti-inflammatory drugs have side effects. And one of the concerns is that if you reduce the inflammation in the lung, yeah, you're saving the patient, but you're compromising their ability to fight off the infection, the actual virus, because they need the inflame. It's also important for fighting infection. So with this therapy, you can actually target just the lungs without affecting any other body organ which is involved in fighting infection. So the inflammation can be still used in other organs. So to sum up, this technology can end the global pandemic, not only the present, but any future ones, and it can do so immediately. That is what this type of therapy could achieve if it's implemented. So, to the next question, why is this not being done? And I'll just give you an anecdote of a little anecdotal response to that. So here, me, in my small person, what have I done? I got these results starting in mid-2020. And since then, I read 7 grant applications. I contacted 10 LED device retailers. I contacted 3 medical device manufacturers. I told prominent scientists in my university and everyone I knew in the scientific community. I contacted doctors working with COVID patients, and I mentioned this work to journalists that I know. So I want I'd like to give you a distillation, just a synopsis, a brief synopsis of the response that I got. which is this. I'll give you a few seconds to enjoy this slide. This is the response to my COVID miracle cure. Ivermectin. Sure, I'm sure. It's science. Heat and light kills COVID. So, oh, and of course, the clinical trials for bleach. So I think the message I want to leave, I'm almost done. So the message I really want to get across, and I'm very passionate on this. And I think this is the consensus I also got from the doctors, the orthopedists who were doing these preclinical trials. This technology will never become mainstream without controlled, professionally conducted clinical trials. Until you have the proof, it will never, nobody is going to believe it. They don't want to believe it. It sounds fictional, it sounds fantastic, and they're not going to try. And I would go a little bit further here. I think that's rightly so. It should be proved. It has to be proved. So why isn't it being proved? Okay, well, what's the problem? I think we hit on this at the last session with Mike Camplin's talk. First of all, the device manufacturers, they're not willing. It's a big financial risk. First of all, it's very expensive to do these things. The design, it's not, you know, there's so many rules about how to do the design, so that costs. It would require testing of the device, which means many more clinical trials. It requires licensing. Nobody is going to ever use this kind of therapy in a hospital in intensive care unless it is FDA approved for this purpose. Who's going to take the liability? Nobody. It has to be licensed. Secondly, for marketing, the problem is risk. First of all, the pharmaceuticals. I'm sure they will not be embracing this with open arms. They have many ways to limit marketing. And of course, another consideration, I'm sure, with a lot of these companies is how long will the epidemic last? You know, hopefully we're at the end of it, but it will take, to get to the point of having something to market, it take another year. Will the epidemic, will it be relevant? So I have, so my suggested solution, I mean, what I'm sort of trying to pursue is to attract venture capital, not so much by proposing for the COVID epidemic, because again, we have these concerns here, but by proposing additional, you know, if it works for cytokine storms people, it will work for many things. It will work for anything where there's underlying inflammation. You need to perhaps do some more lab tests. The dose must be much better defined, but it should work. So this could be a lure for venture capital to propose alternate applications. However, and I have, I and my colleagues mostly who have been involved in these discussions assure me that to get significant funding, it will not be possible unless the initial clinical data is obtained. They're not going to fund it until you show me it works, and then, okay, maybe the money will come. So my conclusion at this point has been, up until now, is that the initial clinical studies, I mean, they must be paid for by private resources. I have tried government grants, incidentally, with absolutely zero luck. So I don't have any hope there. At least, but so private resources more likely, you know, shake up your friends. I mean, I'm ready to sell the farm. I'm not quite sure I would be enough either. So I think we're in a situation where there is an activation energy that needs to be overcome, and this will have to be done by private financing. So I'll let you know a bit about what I've done so far to try to help make this happen. First of all, I've established a partnership with the 4 laser specialists who did the clinical trials. I contacted them. They're on board. They're very enthusiastic. They said their problem is money. There is no money. But if I find the money, they will participate in supervising the trials, establishing the protocols, and providing any kind of advice and support. So they're on board. I'm continuing basic research on the signal, obviously. Furthermore, I have had a device manufactured for testing and tested it that can be used immediately for clinical trials. This is not the final device that would be put on the market, but it is FDA. It's listed by the FDA already, not for COVID, but for other purposes. I've had it modified to my specs. So a device exists already for testing. I've established a partnership with a startup, and I believe the CEO is present on this call, who are interested in the technology, and they are going to provide some of the starting capital, but not all. And the most important thing, I think I've identified and obtained a quote from a competent CRO to actually conduct these clinical trials according to FDA standards and regulations. So they are ready to go. They gave us the quote. They are ready to go tomorrow. And finally, I successfully raised a proportion of the capital towards these clinical trials. And the next step, of course, is going to be to find the remainder of the financing. So now I want to leave this, especially this group here, with a final thought. You know, it's not just about the epidemic for this group, it's about something else that I wanted to mention. And let me give you an example of a technology, a recent technology, which is the BioNTech mRNA technology, which you all know about, invented and promoted by this husband-wife team. So before the pandemic, this is what was said about this mRNA vaccine technology before the pandemic. The wider scientific community was dismissive. Capital markets and large companies were also unconvinced, as Financial Times. After the pandemic, well, it's history, of course. This biotech Moderna vaccine is now mainstream. And as these guys said, there is no possible way that they could have gotten this technology into the mainstream if it had not been for the use that was made available through the pandemic. And I think as regards the Guy Foundation, I believe, since my closing message, I believe there will never be a better opportunity than right now to achieve mainstream standing for a quantum therapeutic method in medicine than to apply what we know now towards solving this COVID epidemic. This will never happen again. It will have if it works to the extent that preliminary experiments suggest, it will have a huge impact, and newspapers will be writing about it. It will become public, social media, everything. It will become visible. It will move from the fringes into the mainstream. And I think this is an opportunity which should be seriously considered by this body. And I end here with thanks to my many colleagues and collaborators. The work I presented now was mainly done by Blanche Aguida and Natalie Jordan, who's here in the audience, also a postdoc, a graduate student, Marut Puam. Blanche is a postdoc. Natalie is a lecturer. Marut Pong is a lecturer. Rachel Sherrard was my collaborator in another lab at the university, who was actually the person who introduced me to pulse electromagnetic fields. I'd never heard of them in human treatment before. So, and also my wonderful colleagues from SIT Research, who have helped us with the pulsed field equipment and studies. Thank you.